It's a pleasure to be here, as always, as every year. Today I will present some of our recent work about network data fusion and how we utilize certain techniques and build certain techniques to, within the same framework, do several things, including patient stratification, biomarker discovery, and to personalize treatment. And this is an overview of today's presentation. Basically, the main idea is that medical systems or biological systems consist basically of a complex world of interconnected entities that we need to mine to discover new biological and medical information. So this is an overview of today's presentation. I will first give a brief introduction, talk about the data and the main challenges, and then I'll present a couple of examples of novel methodologies. In 20 minutes, I don't have time to present much, but basically new methods how to mine these interconnected data and how to mine them at two levels, at the molecular levels of cells, so where we have genetic interactions, protein interactions, metabolic interactions, and so forth, and also at the second level, which is patient-centered data integration, where we want to find out more about disease, symptoms, drugs, and so forth, and to use that to personalize medicine. And in the end, I'll point to some future directions, and hopefully we'll have time for comments and questions. All right, so let's start. We've witnessed an increasing amount of large biomedical data. This is an illustration from our proteomics review paper that appeared in February this year. So we have lots of genomic, epigenomic data, transcriptomic, proteomic, metabolomic, phenomic, exposomic, metagenomic, and so forth. There's like lots and lots of data building up. They're available in these standard databases that we all use, and new databases are appearing, and of course healthcare systems, hospitals, and pharma have much more than that. The idea here is that each data source in isolation is just a different lens through which we look at the same system. And it just describes a specific aspect of the cell or of a patient. And it is all of these data together that we need to look at to get the full picture of the phenomenon we are studying. Okay, so in terms of the cell, we have various types of data such as transcriptional regulation, protein structure, protein-protein interactions, genetic interactions, metabolism, and so forth. We have various types of ontologies such as gene ontology, disease ontology, and so forth. We have patient phenotypes, we have drugs, and we can build various networks on drugs. For example, drugs can be, small molecules can be nodes and their chemical similarity edges and so forth. And of course we have model organisms from which we are transferring information to human and so forth, and these are all just some of the different views of the system that we are studying. And we are currently at a turning point in bioinformatics and medical informatics where these technological advances have yielded large amounts of these data that are posing interesting and important problems to computational scientists, and without computation we cannot really move much forward, and our computational advances have begun to contribute to understanding of disease and understanding of pathogens, aging, and so forth, and having therapeutic effects and healthcare benefits, and we've seen this area booming. Now, to analyze these data, what do we need? Why isn't it trivial to analyze these data? So we've seen over the past few decades that genetic sequences have revolutionized our understanding of biology, medicine, evolution, and so forth. Many of the problems dealing with sequences, with aligning sequences, comparing them, and so forth, fall into the category of so-called computationally easy problems, meaning that given the input, given the size of your input, you can get exact answer in polynomial time of the size of that input. But dealing with large network data is much more challenging, and many of the problems that we are dealing with, many of the questions we asked about our world that is interconnected, fall into the so-called category of computationally intractable problems. NP hard, NP complete, which means that given all the time of the universe and given all the compute power, you cannot exactly solve these problems on your large data sets. But we still want to solve them, and our only way to solve these problems is to design approximate solutions, so algorithms that solve these problems approximately. And one such example is comparison of large networks. So basically, this is NP-hard. Stephen Cook has proven that so-called subgraph isomorphism problem is NP-hard a long time ago in 1971, which means that we cannot exactly compare or align large networks. But we still need to do that. We are still doing that, and we are devising approximate ways or heuristic algorithms to do so. So the challenges are twofold. One is extract as much information as you can out of each of these layers individually, but then also fuse these layers to see what they collectively tell you, okay? And we want to do that in a principled mathematical way. And in this particular case, that is patient-centric, where we have our patients, we have their somatic mutation profiles, we have diseases they have, we have drugs that they've received, but we also know which drugs are prescribed to which diseases, we know which genes are mutated in which diseases, we know which drugs bind to which gene products, to which proteins. We have gene ontology, we have disease ontology, and we have all sorts of layers of molecular interactions, such as gene co-expression, metabolic interactions, genetic interactions, protein-protein interactions, and so forth. We can have various types of networks on drugs, such as, for example, drugs could be nodes, and side effects could be edges, or chemical similarity could be edges, or if they're prescribed for the same disease, it could be an edge, and so forth. Okay, so how do we fuse all of these data first in a unified mathematical framework that we can compute on and extract information out of this data? All right, now let's go to the second part of my talk. As I said, I will just give two examples of how we mine this interconnected world of biological and medical entities. First, only at the level of molecular networks, and then we use these to do this patient-centric, patient-centered data integration or data fusion. And I will just give an example in cancer, but we are doing similar things for cardiovascular disease, diabetes, arthritis, polycystic ovarian syndrome, and so forth. But in 20 minutes, I'll only talk about cancer example. Okay, now in terms of molecular networks, this is an older work that we've been doing in my group over the past 15 years or so. And basically deals with large networks, often protein-protein interaction networks, but whatever your entities are, they are represented as nodes and their interaction as a link. And then presented with another network, you want to find out whether those networks are similar, how similar they are. We know we cannot know exactly how, whether they're the same or not when the data is large. So we do simple things. For example, we compare if they have the same number of nodes, if they have the same number of edges, if they have the same degree distribution, meaning that the degree of a node is basically the number of edges incident to that node. But actually very quickly you can come up with these examples where you have networks with the same number of nodes and edges, and in each node, in each network, each node has the same number of links, but they're very different. For example, this one is disconnected, consists of triangles, and this one is a large cycle. So this is not enough. And quite a while ago now, 12 years ago, I published a paper in bioinformatics on graphlets. And the idea is that these graphlets can be viewed as building blocks of large networks, as Legos of these networks. And we are asking whether we can figure out from the patterns of connectivity whether we are dealing with protein interaction network or some other type of network, or in Lego terms, whether based on how we assemble these Legos, whether this is a pumpkin or whether this is an airplane. So basically it is how these small substructures, these small subnetworks or graphlets fit together that is very data set characteristic. The dependencies within data, so basically the number of triangles in the data will impact all of these other graphlets that have triangles, but we shouldn't shy away from this. This is good because we can exploit these dependencies because they are data set specific, and then we can learn much more about our data, both at the level of the entire network, but also at the level of individual wiring patterns around particular entities, for example, around particular proteins in protein-protein interaction networks. I won't have the time to talk about graphlets now, but many of you have heard me speak about that for some time. I'll just like to point out that in the issue of Science from 2 days ago, my postdoc, Noel Malodognin, and I have an insights or perspective paper on network analytics in the age of big data. where we highlight these graphlets because Yuri Leskovec had just used them in a very interesting and smart way, and he published in the same issue a paper how he utilizes them to discover hierarchical principles in various network data. Just as a highlight, basically the idea that we've been working on and we've shown over the years is that these wiring patterns around proteins in protein interaction networks are not random, and they're very characteristic of biological function, membership in protein complexes, subcellular localization, tissue co-expression and involvement in disease, and that we've used just basically these wiring patterns to find new members of melanin production pathways that were phenotypically validated with collaborators at UC Irvine School of Medicine. And also more recently, we've shown how these wiring patterns around proteins are characteristic to particular Go terms, and how that's conserved through evolution. But and we generalized everything to directed networks as well. So if you want to study, for example, metabolism, you can do that using directed graphlets. And actually, all of that is applicable to network dynamics. And we have tracked network dynamics both in the undirected and in the directed case. But let's now shift gears and go to this patient-centered data integration. And its application in precision medicine. And I only have time to talk about one example, and this is our recent paper in Pacific Symposium of Biocomputing this year. So we took 353 ovarian cancer patients from TCGA. So we have three objects. We have patients, we have genes, and we have drugs. And we have somatic mutation profiles of all of these patients that we took from TCGA. Then on genes, we have various types of molecular interaction networks that we also took from public databases. And in this particular case, we took protein-protein interaction, genetic interaction, and metabolic interaction networks. Then we took drugs from a drug bank and built a drug chemical similarity network on them. And we also have information on drug-target interactions, also from drug bank. So within this same framework, we will do patient stratification, driver gene prediction, so basically prediction of new genes that drive progression of this particular cancer, and we will propose drugs that could be repurposed. And basically drug repurposing is one of the big pushes today by the pharma industry and also by the national healthcare system because of the time and cost savings for basically, if you can repurpose already existing drugs that you know that are good, not toxic and so forth to different patient groups, that would be of great benefit. But within the same framework, we would do these three things. How do we do that? We want to simultaneously cluster or co-cluster three types of objects: patients, genes, and drugs. Okay, and how do we do that? Okay, so the motivation for our method comes from non-negative matrix factorization, which factorizes a homogeneous matrix. This is your data that is homogeneous of one type into two matrix factors. And this is actually, this method is an alternative to principal component analysis and vector quantization. And the theoretical foundation for this comes from the fact that this factorization is equivalent to a relaxed form of k-means, where F contains the centroids of the clusters and G is the cluster membership indicator matrix. So to which clusters do the data points belong, and K is the number of clusters, which is small, which is much smaller than the size of your data, okay? So this is for homogeneous data, for data of the same type. We factorize them into two non-negative matrix factors. But we are dealing with heterogeneous data. We have patients, genes, and drugs. And this is why we have to use a non-negative matrix 3 factorization. So there are going to be 3 factors here. And it's going to be penalized, or also called graph regularized, because we want to fuse inside the information coming from molecular networks and from drug chemical similarity networks. So how do we do that? Okay, so first let's just focus on patients and genes. So these are somatic mutation profiles. So this is this matrix R12. We will factorize it into 3 matrix factors. This is basically your patients. K1 is the number of patient clusters, which is going to be much smaller than the number of patients. Then there is this matrix here of genes, okay, and K2 is the number of gene clusters, again, much smaller than the number of genes. So these are your cluster indicator matrices, and this is a compressed version of this matrix that you are approximating. Okay, and basically you can view it as kind of containing latent structures, smaller dimensional structure of your data. Okay, so as I said, penalized because we'll be integrating topologies of molecular and other networks. Non-negative because we want to have cluster interpretation. And it's three-factorization because we want to co-clusters patients and genes. But this is just patients and genes. We also have genes and drugs, and we do the same. We decompose R2-3 into these three matrix factors, okay? So R1-2 into three matrix factors, R2-3 into three matrix factors. But here we are sharing this matrix factor G2 of genes, and this ensures the fusion of your data. And I'll come back to that later. Now, again, just rewritten, R1-2 is the product of these three matrix factor, R2-3 of these. We are sharing this red G2 matrix factor. And now, how do we do this? How do we approximate these R1-2 and R2-3? Well, we are solving another NP-hard continuous optimization problem, which minimizes the distance between R1-2 and the product of these matrices, and R2-3 and the product of these matrices, okay? So we're finding the minimum, but this is under the guidance of these constraints that come from the layers of molecular networks and from drug chemical similarity networks. So basically these Laplacians here mean that the physical connectivity will guide your minimization procedure towards a minimum. It will reward connected nodes in your networks, and also we built in these graphlet sort of things, basically that will reward the wiring patterns that are similar around proteins, even when your proteins are not connected. Okay. Now, how do you minimize J? Well, you have to do some mathematics here. You have to derive, here we used KKT optimality conditions, multiplicative update rules. Basically, we start with random matrices, or there are some other ways, but let's say these, you start with random matrices, and then these update rules that you derive that are guiding your J towards your local minimum, of course. You cannot get global one because this is NP-hard, and then you stop when within two iterations the difference is not very big. Okay. And now, just the highlight of some results. So basically, where are you getting your patient stratification, driver gene prediction, drug repurposing from? So for patient stratification, you're reading it from this matrix that contains clusters of patients, and we found within these 353 patients, 3 clusters with statistically significantly different survival outcomes. These are Kaplan-Meier survival curves. In terms of driver gene prediction, again, from the same framework, we are getting this from these clusters G2, okay? These are clusters of genes. So basically, in them, we get these clusters that are statistically significantly enriched with already known drivers, and then we're hypothesizing that inside of them, there are also some new drivers, and then we get validations for that. For example, 40% of our predicted driver genes we validated in the databases that we intentionally didn't use in this fusion to be able to validate how well our approach does. And then when you look at the function of these predicted drivers genes, you see that it also makes sense. In terms of drug repurposing, it comes from this R2-3 here. And over here in this graph, I'd like to show you the power of fusion, of data fusion. Because if within this framework, we are only using drug-target interactions to predict new drug-target interactions, you're not very accurate. These are area under the precision recall and ROC curves, but then as you add molecular networks, somatic mutation profiles, and so forth, as you add all data, you're getting progressively more and more accurate, and basically this illustrates the power of the fusion and why you need to do that. And the beauty of this methodology is that you can add as many entities as you want, as many data as you want, or you can subtract and find out how much your model degrades, or what is the contribution of each data source, how much information is within each data source. Okay, and again now in terms of drug repurposing, 37% of our predictions we validated in other databases that we did not use in our fusion framework. This is another example just to illustrate what we are doing. So over here, this is ongoing work with Professor Stebbing's lab at Imperial College. And these are actual patients that they have, about 45 patients, cancer patients. This is an illustration of cancer heterogeneity, where over here we have patients, and we see, and the genes that are mutated in those patients. And we see that each patient usually has a very small number of mutated genes. There would be an occasional patient with lots of mutations. Now, in terms of genes, you see that genes would be, each gene would be mutated in a very small number of patients, except maybe TP53, that is mutated in about half of our patients. So over here, we have patients, drugs, genes, their actual mutations. We have therapies that these patients received. We have, of course, drug-target interactions, all sorts of these other layers. And this is the fusion framework where we are approximating these R12 by these three matrix factors, R13 by these, R23 by these, and you can see how we are sharing R1, we are sharing R2, we are sharing R3, and this really empowers the fusion when you can do this, because then even for a small number of patients you can get your results. And basically we want these predictions here, repurposing of known drugs to the patients. All right, now this is where I'll stop talking about this patient-centered approach. I can just say that we've used a similar approach to do other things. For example, disease reclassification. Disease classification, as we know, it is about 100 years old, and it's based only on the symptoms and the most affected organs, because 100 years ago, we didn't have all of these molecular level data. So the question now is whether it should be rewired in the light of the new molecular network data. And over here, we have genes and all sorts of molecular interaction networks on them. We have drugs, we have drug interactions, we have Go terms, we have disease ontology terms, and basically now we have 4 objects. Previously, in the previous example, we had 3, and then we are fusing all of these matrices and connectivity patterns to figure out the disease, whether disease ontology needs to be rewired. And what we find is that basically by looking at only these wiring patterns, we recover 80% of disease ontology only from the network data. So for the other 20%, I would suggest that we use the other data that was originally used to come up with this disease ontology. But definitely, as we all know, there will be room for improvement here, because we already know that seemingly unrelated diseases are caused by the same molecular mechanisms, and vice versa. We have examples of that. And just as the last highlight of the result, how this methodology is powerful, basically, we did multiple network alignment with the same methodology. We just published that in Bioinformatics earlier this year. And we know, as I already said, even just aligning 2 networks is computationally intractable. Aligning more than 2 networks becomes a much harder problem. So over here, we are aligning human protein interaction network with 4 other model organisms. Across, we have sequence similarity between these proteins, but then we're also integrating molecular networks, topologies of molecular networks in the same framework that I show you. This is minimization problem that I've shown you. And then this is just an illustration how you share matrix factor. This is just from Homo sapiens to others, but this is a full graph. It was just too messy to include it all. And then once you obtain, even once you recover these R12, R13, and R23, and so forth, then you still are left with a k-partite matching problem, in this case 5-partite matching problem, which is NP-hard, so we have to do it heuristically. And my time is coming up, and I can just tell you how, both in terms of biological process and molecular function, integrating topologies gives you clusters across all 5 species that are more functionally consistent than the sequence only, the dashed line is sequence, and we get clusters over 5 networks, unlike other methods. All right, so I probably won't have time for future directions. Basically, the idea is to use these and build a recommendation system for several diseases, and I am doing this with my collaborators at UCL. Mining data will stay complicated. These problems are to stay with us. They're due to large sizes, heterogeneity, highly dimensional data of growing complexity, which are noisy, dynamic, at different time and space scales. And the data complement each other, but we need new methods to mine them. And I think here I will skip and just thank my funding agencies and my group members. And thank you very much for your attention. I can answer a couple of questions.